Students, we are going to learn about Pinus, which is in the syllabi of Semester 3 Botany students, Paper 1, Unit 3. Pinus is commonly known as pine. You might have seen this tree when you have visited to the mountain slopes in Kashmir, Himachal Pradesh, Uttaranchal, don't you? Yes, those tall conical trees on the mountain slopes, they are Pinus or pine. We are going to learn in terms of morphology of vegetative parts and reproductive parts, anatomy of stem, leaf and root, its reproduction and its life cycle. Let us first know its systematic position. It comes under kingdom Plantae and subkingdom Embryophyta, group Tracheophyta or Spermophyta, subgroup Gymnospermi, division Coniferophyta, class Coniferopsida, order Coniferales, family Pinaceae and genus Pinus. Pinus is the largest genus of the family Pinaceae with 100 to 105 species spread worldwide. It is found in temperate to subalpine regions on high altitudes. Let us know its distribution in India. Almost 6 species have been reported from different parts of our country. It occurs in Himalayan regions, hill slopes of northeast India like Kashmir, Himachal, Uttaranchal, Assam, Manipur, Andaman and Nicobar Islands. Common Indian species are Pinus roxburghi, also known as Pinus longifolia, popularly called chair pine, Pinus giardiana, known as Chilgoza pine. Students, you might have seen those Chilgoza dry fruits. Yeah, they are obtained from Chilgoza pine or Pinus giardiana. Pinus wallichiana, known as Pinus excelsa. It is also called silver pine, silver fir, blue pine or cal. Pinus insularis or Pinus khassia, popularly known as khasi pine, Pinus mercusi or mercus pine and Pinus armandi. So, these are the Indian species of Pinus. Now comes to the external morphology of Pinus tree. Pinus, the main plant body of Pinus is sporophytic, which is evergreen, perennial and xerophyte. It has a conical or pyramidal appearance, or we can say excurrent habit, just because of its spiral growth of lateral branches. Its plant body is differentiated into well-developed root, stem and leaf. Taproot, the taproot of Pinus, it is the main root system. It has lateral roots which are well developed and it helps in anchorage and absorption of water. You know, because Pinus are found on hill slopes, so it requires a good anchorage and good absorption of water system. On the lateral roots are found ectotrophic mycorrhizal fungal association, which acts as a symbiont. Now comes to the Pinus stem. Pinus stem is erect, aerial, cylindrical and branched. It is woody. Older stem has bark which is scaly, brown and rough. Let's come to the Pinus stem. Pinus stem is divided into 2 branching systems. One is branches of unlimited growth, which are also known as long shoot, and the other is branches of limited growth, which is known as dwarf shoot. Both the shoot have scale leaves, which are known as cataphyls. Pinus leaves, it has juvenile leaves or primordial leaves which are found only in its young stage. Otherwise, we can say that there is a dimorphism found in the leaves of Pinus. That means it has 2 types of leaves. One is called scale leaves, other one is foliage leaves or Pinus needle. Let's know about scale leaves of Pinus. They are non-green, brown, membranous, or we can say very thin, small and brown in color, found on both long and dwarf shoot. Then the foliage leaves or Pinus needle. They are green, photosynthetic, acicular, that means they are needle-like, and that's why we call Pinus needle instead of saying the foliar leaves of Pinus. They are pointed also. When we talk about the needles of Pinus with the dwarf shoot, it is known as spur. There are different types of spur in Pinus which are based on number of needles found in it. Like if there is one needle in a spur, it is called as unifoliar or monofoliar spur. If there are two needles, it is called bifoliar. With three needles, it is trifoliar, 4 is quadrifoliar, and 5 are pentafoliar spurs. Now we comes to the anatomical part. Anatomy, that means internal structure. So we'll first learn about the internal structure of root. The outermost layer of the Pinus root is called piliferous layer, which is single layered. Then comes the cortex, which is parenchymatous. After cortex, there is endodermis, single layered, and multi-layered pericycle. The stele or the vascular system, the stele is diarch, triarch or tetrarch, that is xylem is exarch with racine canals. Phloem alternates with xylem, but students, please remember that there are no vessels present in xylem and no companion cells are found in phloem. Secondary growth is just like that of dichotrid. Okay, now we'll learn about the internal structure of Pinus stem or stem anatomy. The outermost layer of the stem is epidermis, which is single layered, but remember it is cuticularized. That means it is meant for retarding transpiration because it is a xerophytic plant. Next comes is cortex, which is sclerenchymatous, that is also a xerophytic character. Racine canals are found in the cortex of Pinus stem. Then endodermis and pericycle, which is not very distinct. Vascular bundles are called conjoined, collateral, endarch and open. Primary cambium is found with medullary rays. Xylem is present, which is exarch, but vessels are absent, and phloem, in which companion cells are absent. Pith is parenchymatous but poorly developed. Like other dicot plants, the secondary growth of stem is similar in that of Pinus also. Now when the secondary growth takes place, the cambium get activated and it forms secondary phloem from outside and secondary xylem inside. The growth is so massive that it forms a bulk of secondary tissues, and that's why the stem of Pinus get broadened. You see many rings in the Pinus stem. What are those rings? They are called Anvil rings, and they are formed due to the secondary growth and the activity of cambium. Spring wood has wide tracheids, while autumn wood has narrow tracheids. That is why you see those broad rings and narrow rings. They are annual rings of Pinus. They are very important and they shows the secondary growth. Racine ducts are present both in the primary and secondary wood of Pinus. Now come to the anatomy of Pinus foliar leaf, that is called needle. It is very important because you people get Pinus needle in your practicals also. That is very simple. You cut a thin section of the needle, double stain it, mount it and see with compound microscope. What you see? It is a triangular structure with outer cuticle, then the epidermis. It has sunken stomata, presence of cuticle and sunken stomata. it shows as xerophytic characters. Then comes the sclerenchymatous hypodermis. Mesophyll cells are present, but they also show infoldings. It is an important character of Pinus needle. Then comes the endodermis, which is single layered, and pericycle, which is multi-layered. But what you find is the sclerenchymatous T-shaped transfusion tissues. It is also a xerophytic character shown in Pinus needle. Vascular bundles are usually 2, conjoined, collateral and endark. This is the structure of Pinus needle. Now we come to the reproductive part of Pinus. Pinus is a heterosporous, or we can say monoecious species. It has male cone as male reproductive organ and female cone as female reproductive organ. Male cones are more frequent on lower branches and they are more in number, while female cones are found singly or less in number and usually on the top of the branches. When we come to male cone, it is made up with microsporophylls, which has microsporangia filled with microspore. Let's talk about female cone. It is made up of megasporophyll, which has megasporangia and megaspore. This is how male and female cones are formed. Yes, let us come to the structure of Pinus male cone or male strobila. It is born in clusters, but these are small, yellowish green when they are young, but become brown or dark when they become mature. The longitudinal section of Pinus male cone shows that there is a cone axis on which microsporophylls are arranged spirally. Microsporophylls has sac-like structure, that is microsporangium. Inside microsporangium are found microspores, or we can say pollen grains of Pinus. Let's talk about the development of microsporangium of microspore. It is eusporangiate type of development. What do you mean by eusporangiate development? It means it is formed from a group of cells. Inside microsporangium are microspores or pollen grains. Pollen grains is a haploid structure which has intine and exine, and exine forms balloon-like wings which helps microspore or pollen grains in pollination. So, it is unicellular and haploid. Now, we will talk about the female cone of Pinus. It is oval to elongate, very beautiful. You might have seen these female cones in the drawing rooms as a part of arrangement. Yes, they are larger, developed singly and woody. There is only a 3-year life cycle of Pinus female cone. So, 3rd year cone is a matured cone which takes part in the reproduction. Now, when we come to the structure or the longitudinal section of a female cone, it is made up of many megasporophylls which are arranged spirally on the cone axis. Megasporophylls are made up of 2 types of scales, that is ovuliferous scales and Bragg scales. Ovuliferous scale are flat, triangular, known as apophyses. They are bigger and woody, while Bragg scales are small, membranous. They get enfolded and helps in fertilization and pollination. On the ovuliferous scale, at the base of it has 2 ovules. The ovule of Pinus is orthotropous unitegmic. That means it is made up of 1 integument. These integuments are made with 3 layers. Outer and inner layer is fleshy, while the middle layer is stony. The upper part of the ovule is not covered with integument and it forms a pore or opening known as micropyle. Inside micropyle is pollen chamber and then the inner part of the ovule Okay, now this is the structure of ovule. These are the sporophytic structures of Pinus. Now we are going to learn about the gametophytic phase. So, the gametophytic phase starts with the development of gametophyte with the germination and of pollen grain and with the formation of megaspore in the female cone to form a female gametophyte. Microspore or pollen grain, it is the first structure of male gametophyte. So, first we are going to learn about the development of male gametophyte. The development of male gametophyte from the pollen grain takes place in two stages, that is the development of male gametophyte before pollination and the development of male gametophyte after pollination. Let us talk about the development of male gametophyte before pollination. The pollen grain inside the microsporangium gets divided and forms a 4-cell structure. Up to this 4-cell structure, it is called the male gametophyte. This male gametophyte get transported from male cone to the female cone through pollination. You have remembered that those balloon-like wings, they help these pollen grains for the pollination. At the time of pollination, a transverse slit is formed in the microsporangium of male cone, and these pollen grains in the form of 4-cell structure of gametophyte comes out, and they are so numerous in number that it is like yellow colored cloud in the sky, which is known as sulfur spring. These pollen grains get distributed all over the area and reaches the female cone, where there is ovule intact in integuments. When this male gametophyte in a 4-cell structure reaches female cone, the micropyle end of the female cone or the ovule, it starts discharging a sticky substance through which the pollen grains get stuck. This substance gradually becomes drier and the pollen grains starts coming inside of the micropyle. Then they come into the pollen chamber and there they get developed into the rest of the stages of male gametophyte. Okay. Now we will switch over to the development of female gametophyte. You have seen that the female gametophyte is in the form of ovule, but the inner side of the ovule get divided into megaspore, and this development is also eusporangiate, that is, it is formed from a group of cells. Now it forms megaspore, and megaspore develops into archegonia, and archegonia has egg cells. So this is how the female cone get the development of female gametophyte. Now your pollen grains are ready for fertilization and they are in the pollen chamber. We will switch over to the rest of the development of male gametophyte. In the pollen chamber, a pollen tube is formed which comes inside and reaches the archegonia, and the pollen grains get divided into many cells in which there are 2 male nuclei or 2 male gametes. One of the male gamete enters into the pollen tube and it fertilizes the egg of the female gametophyte. Now the fertilization takes place and embryo is formed. This embryo gradually forms the seed inside the female strobilus or female cone. When it is get matured, the seed come out and spread over the nearby area. The germination of seed takes place through apical type of germination, and first of all the juvenile leaves are formed and the young root or radicle goes inside the soil and the plumule comes outside. Juvenile leaves are soon they get degenerated and forms or develop into a shoot. This is how the reproduction and life cycle get completed. I hope students you have learned the whole process and you found it interesting. Thank you.